All right, I want to show you what you're going to be doing in an experiment on enzymes. This is biochemistry number 6, and you're going to run a enzyme catalysis lab. So catalysis means to break something down. Your substrate is H2O2, hydrogen peroxide. Hydrogen peroxide gets broken down into water and oxygen gas. So the enzyme that we're going to use is called Catalysis, and you can find Catalysis in any living cell. It's one of the really abundant enzymes. So we're going to use potatoes, and we're going to grind up the potatoes and use this enzyme from the potatoes. Now, this reaction, hydrogen peroxide being converted into water and oxygen gas, you can see the effects quite a few ways. If you just let it go, you will see bubbles of oxygen form, but it happens so slowly. If we just stared at it, it wouldn't happen. Once you add the catalase in, you speed up the reaction and you can actually watch it bubble. Bubbles is not a good way of measuring though, so we're going to use a titration. So if you remember the way a titration works, you fill up a burette with an indicator. We are going to use KMnO4, which is called potassium permanganate. And the amount of potassium permanganate that we use is directly proportional to how much H2O2 is present. So we're going to be looking for disappearance of substrate. If you think about it, there's a lot of things you could do. You could compare how much did I start with of my substrate and how much did I end with, or you could measure how much product have I made. It's difficult to measure either one of our products, water and oxygen, so instead we can use this indicator. It binds to H2O2 and I should be able to measure how much remains. So what you're going to do is you're going to set up beakers. And in every beaker, you are going to add H2O2. Well, you're going to have a control. So I'll call this one my control. And for the control, we're going to add water instead of adding an enzyme. To the rest of them, I'm going to add H2O2 and I'm going to add an enzyme. And the difference between each of these is how long I'm going to let the enzyme work for. So I'll let the enzyme work for 10 seconds, and 30 seconds, and 60 seconds, and so on. You'll keep going. You'll have more containers than this. So my first question to you is, how can I get the reaction to stop? Because if I only want it to work for 10 seconds, I need to have a way of stopping the enzyme. So think about it for a minute. I'm hoping that you're coming to the conclusion of I could denature the enzyme by adding an acid. So my acid will be ready and I will add it at 10 seconds. If you don't have it ready in advance, there's no way you're going to get it ready in 10 seconds. So this lab, you have to be a little bit tricky and prepared each second of it so you know what you're adding next. Once I add the acid, I've stopped the reaction, I can set it aside, and I can work on the next one. So this one, I add the H2O2, I add the enzyme, but I let it work for 30 seconds. Add the acid, stop the reaction. So after a while, you're going to have all these containers, and you're going to want to figure out how much hydrogen peroxide did I actually use up. So you have to create for yourself a baseline. A baseline is going to tell you how much H2O2 you had at the start. Once I know how much I have at the start, so for example, let's say I had enough KMnO4, I used up, let's say 15 milliliters of KMnO4. These are directly proportional, so I just want to know how much of this I have used up. Maybe after 10 seconds, what do I expect? I expect less of this needed. So let's say after 10 seconds, I only need 13 milliliters of KMnO4 because some of the H2O2 is gone. Some of it was decomposed by the enzyme. After 30 seconds, what should happen to this number? It should go even lower. Maybe now I only needed 10 milliliters of KMNO4. So you get the idea. The longer you let the enzyme work, the greater it should work. Now, there is a maximum rate at which an enzyme can work. So if this is my graph, and I am measuring how much H2O2 is decomposed over time, I can have my 0, my 10, 20, 30, 40, 50, 60 seconds, and I should be able to see, at the beginning I have none decomposed, so I should be able to see my rate go up, but at some point it's going to do this and it levels off. So when an enzyme levels off, that is telling me that my enzyme is at maximum rate. When it's at its maximum rate, it's working as fast as it can. You could keep adding more H2O2, but it can't work any faster than it is. All right, if you have other questions, please make sure that you come ask me.